In a publication from the TCGA Research Consortium, it was reported that in glioblastoma, mutations in the genes EGFR, NF1, and IDH1 and PDGFRA define classical, mesenchymal, and proneural subtypes and identified gene signatures for these subtypes. Let's look at how mutations in these genes correlate with gene signatures. We will create an omic data query of DNA-seq somatic mutations. You can enter one or more genes in the gene search. We will generate a separate query for each of these genes. Under Custom Query, we will find the mutation status for each. Let's hide missing samples. And filter only for glioblastoma. Now let's switch to the Group Association Clinical Significance view. Change the grouping to one of our genes' somatic mutation statuses. For each gene mutation status, glioblastoma subtype associated gene sets from TCGA gene signatures were identified. To explore these, switch to Numeric Clinical Variables. Under the Clinical tab, since we don't want to view all charts, first uncheck all, then search for GBM. Choose the TCGA marker paper subtype gene signature groupings. Four charts will be shown, grouped based on our somatic mutation status for one of our genes, and each sample's similarity to the defined gene signature is plotted. We can scroll through to find that IDH, somatic mutation status, tends to correlate with a proneural subclass. In contrast, NF1 somatic mutation status tends to correlate with the mesenchymal subclass. Finally, EGFR status correlates with the classical subclass gene set.